Hi guys, and welcome back to another immunology session for Bio 265. Today we're talking about video 3.2.1.3, which is going to discuss B cell activation. To kind of get started, we just wanted to revisit a topic that we've discussed in the last two videos. We wanted to just repass and go over what happens and where our B and T cells actually originate from. Again, our B and T cells originate from lymphoid progenitor cells that are found in the bone marrow. However, unlike T cells, B cells will actually stay in the bone marrow where they will go through a process of maturation similar to T cells. Likewise, they have to be negatively and positively selected. In order to be positively selected, a B cell must be able to produce a functional BCR, which in this case will be IgM or IgD antibodies. These functional BCR then has to be able to react and bind to a foreign antigen. And if it can do so successfully, then we're good to go. To be negatively selected, we have to make sure that these BCRs also do not recognize self-antigens. If it doesn't, then it will actually be able to be completely processed and move out of the bone marrow into the secondary lymphoid tissues. Just to give a brief overview, we want to look at these images real quick just so that you guys know what we're talking about in the videos. Here on the right, we've got our B cell, and our B cell has our BCRs, which are our IgD and our IgM antibodies, just as depicted over here. Our B cell also has MHC1 proteins and MHC2 proteins on it. In order for a B cell to be fully activated, we're going to look at a lymph node. As previously mentioned, if it is a mature naive B cell and has actually successfully achieved maturation in the bone marrow, it'll move to the secondary lymphoid organs. In this case, we're looking at a lymph node. At this point of the infection, we're about 8 to 10 days into it, and our pathogens have actually entered into our lymphatic system. So it is flowing through our lymph as we speak. This is good because then it means our B cells can actually get activated faster. So if there's a B cell that is actually able to actively identify our pathogen, the BCRs will actually bind to it, which will lead to phagocytosis of our pathogen. The pathogen will then be degraded, and then the antigens will be presented on our MHC2 molecules. If there is an activated helper T cell, a TFH cell, or an activated CD4 plus cell, all of which are the same, just different names, we will be able to actually lead to full B cell activation. But we have to find one of those. So the B cell will hang out in the lymph node, which could take an extra day or two until the correct helper T cell comes along. Once a TFH cell that can actively and accurately bind to this antigen presented on MHC2, we will actually get binding of the TCR of the TFH to the antigen and also the CD4 molecule to the MHC2 protein. This is the first step of B cell activation. The second step of B cell activation is the binding of co-stimulatory molecules between the B cell and the TFH cell. The third step is the release of cytokines from the TFH cell onto the B cell. These cytokines will help determine what antibodies will be secreted next. Once the B cell has received all three molecules and signals, it is then able to differentiate and proliferate into two types of cells. First, it'll form memory B cells, as depicted here up top, and then it'll form plasma cells, which will be our antibody secreting forms. The memory B cells are meant to be kept for later use so that they can actually help prevent infections faster. But as I said, these cells will proliferate and differentiate very quickly to produce as many cells as possible so that we can get as many antibodies secreted as possible. And then these plasma cells in the bottom here that are full of antibodies will secrete the antibodies into the bloodstream to help prevent further infection and destroy any existing pathogens in the body. Just to give a brief overview, there are 5 main antibodies that you need to worry about. We have IgM over here on the left, which can form a pentameric form. We've got IgE, we've got IgG, we've got our IgM, and then we've got our IgA. Other than that, that's pretty much all we have to talk about for B cell activation. We just want to say thank you guys for watching.